Hi, I'm Laura Havers and I'm a PhD student in the Gel Lab at Birkbeck, and it's a pleasure to be making this short presentation in celebration of 100 years since the birth of Rosalind Franklin. I'll be talking today about just one of the methods that I've used as a student in the Gel Lab. I'll be talking about how we use the twin design to quantify the influences of genetic and environmental effects on individual differences in traits. So the twin design is an incredibly simple yet powerful design that doesn't require DNA information. All that it requires is knowing whether twins are identical, so whether they're monozygotic, or whether they're non-identical, so dizygotic. And we derive separate association statistics for monozygotic and dizygotic twins, which reflect the within-pair similarity for a trait or traits. And we use what we know about genetic and environmental relatedness for these different types of twins. And we then fit formal statistical models to our data in order to estimate genetic and environmental influences. So we know that MZ twins share 100% of their additive genetic variation. And we know that DZ twins share on average 50% of their additive genetic variation. We also know that all twins growing up in the same family, like all siblings, share 100% of their common environmental influences, like, for example, the neighbourhood they grow up in, and 0% of their non-shared environmental influences, like, for example, having different friends. The traits or phenotypes that I've been looking at are collectively termed psychotic experiences and negative symptoms. And this is thinking about the symptoms of schizophrenia as more mild manifestations in the general population. So these traits were reported at 16 years and 17 years in the twins' early development study. And in the data set that I use, there's data from approximately 4,500 twin pairs at 16 and 1,500 twin pairs at 17. And I use the twin design to ask about the extent to which genes and environments contribute to individual differences in these traits, and individual differences in these traits reported across age. And the results I'll talk about today are from work that I did primarily as an undergraduate student, and these results were published in 2019. I'll focus here on paranoia, and examples of paranoid experiences are things like thinking that you're being followed and thinking that people are talking about you behind your back. So we found that across the whole sample, the association between paranoia reported at 16 and paranoia reported at 17 was 0.63. And this standardized association statistic, which is called the phenotypic correlation coefficient, represents a substantial association, which is perhaps not too surprising given the short time period. And we found that 42% of this phenotypic correlation was explained by genetic factors, 20% was explained by common environmental factors, and 38% was explained by non-shared environmental factors. And we can go one step further and look at the variance or individual differences in our traits at 17 years. And we can decompose this variance into that which is due to genetic and environmental factors shared with at age 16, and these factors that are not shared and are unique to age 17. The results here are from across all the individual traits that we looked at, and I'll just focus again here on paranoia. So we found that of the variance or individual differences in paranoia at 17, 44% was due to genetic and environmental factors shared with age 16, and 56% was due to unique factors at age 17. And these results are really interesting as we see that even over such a short period of time, we have new genetic and environmental factors coming online to influence differences in these experiences. And looking further into the results, we found that for the stability of paranoia, so of these shared components, the biggest contribution was genetic at 25%. And for change in paranoia, so of these unique components, the biggest contribution was non-shared environment at 44%. And this pattern a pattern of results was generally found across all of the psychotic experiences and negative symptoms domains that we looked at, but with some variability. And this variability really highlights the importance of studying these as separate traits rather than aggregating them into a single domain. So to summarise, both genetic and environmental factors contribute to the cross-age associations across psychotic experiences and negative symptoms in adolescence. And we found that stability was most attributable to genetic factors and that change was most attributable to non-shared environmental factors. And next in my PhD, I'll be incorporating both data collected on these traits at 21 years in early adulthood, and I'll also be investigating the effect of incorporating scores from a general factor of cross-domain psychopathology on the genetic and environmental estimates. If you're interested in this research, you can follow me on Twitter or feel free to contact me via email if you have any questions or feedback. And thank you for listening.